Hello students, welcome to BioSmart Academy. In today's lecture, we will continue our lecture series on structure of proteins. Okay, we already made 3 videos on structure of proteins. So please go and watch that lectures first and came here to watch today's lecture. Okay, so we were discussing about the secondary structure of protein, and we discuss one major type of secondary structure of protein, that is alpha helix. Okay, we discuss it completely, that is alpha helix we already discussed. Okay, we completed the alpha helix part. Okay, there are 2 lectures on alpha helix. Please go and watch that 2 lectures. Okay, that will give you benefit. Okay, now today we will going to discuss about the beta conformations. Okay, and we start beta turns. Okay, so this will be the part 1 of this lecture. Okay, in our part 2 lecture we will discuss beta turn in details. Clear? Now let's start today's lecture. Okay, remember student, these all lectures are very important, and we already are in lecture number 22 of biochemistry lecture series. So we already uploaded 21 lectures, and all that lectures were strictly made according to CSIR net syllabus. So please go and watch all that lectures. Okay, now so let's start beta conformations. See, I already discussed about the alpha helix, and during alpha helix I told you that alpha helix is basically take a helical form. Okay, a helical form, and a rod like axis is there around which it basically just formed a helical like structure. Okay, but today we will going to discuss about the beta conformation. Okay, so in 1951, Pauling and Corey again predicted a second type of repetitive secondary structure. Second type of repetitive secondary structure. Clear? Second type of repetitive secondary structures, and these secondary structures are nothing but these are the beta conformations. These are the beta conformation. Clear? Beta conformations. Okay. So basically, beta conformation is more extended polypeptide chain as compared to alpha helix. So when we discuss alpha helix, we already discussed that alpha helix is more compact in nature. That is, its compactness is very compact in nature. Okay, and we also looked this alpha helix and beta conformation in the Ramachandran plot, and we also seen there that alpha helix is always, the value of alpha helix always range close to 0. Okay, close to 0, either 39, 47, in that area. But when we look the beta conformation, that is parallel beta sheet and as well as anti-parallel beta sheet, we see that the structures are extended in nature, and their values are ranges towards 180 degree. Okay, so we already seen all these things in Ramachandran plot. Okay, if you not watch the lecture of Ramachandran plot, then please visit that lecture. You will find out in our biochemistry playlist. Okay, so basically beta conformations are more extended structure. Okay, please note it down, more extended structure as compared to alpha helix. Okay, and is again defined by backbone atoms. It again defined by backbone atom arranged according to the set of dihedral angle. Okay, according to the set of dihedral angle. Okay, so these are also arranged according to the dihedral angle. Okay, and we discuss about this all these dihedral angle in our Ramachandran plot lecture. Okay, so understood. So beta conformation is another type of repetitive secondary structure. This was discovered by again Pauling and Corey in 1951, and in beta conformation the structure is more extended in nature as compared to alpha helix, and is again defined by backbone atoms. It is again defined by, just note it down, backbone atom like that of alpha helix, backbone atoms. There is no role of side chain in secondary structure. Okay, so it is again defined by the backbone atoms, very important, backbone atoms, and according to the set of the dihedral angle. So this is the basic introduction of the beta conformations. Okay, next. So in beta conformation, the backbone of polypeptide chain is extended. in zigzag manner. Okay, so if we look at the structure of beta conformation, then we find out that in beta conformation the backbone of the polypeptide chain, okay, so if we look at the structure of the beta conformation, we will find out that the backbone of the polypeptide chain in case of beta conformation is arranged in a zigzag manner, okay, rather than helical, which is in the case of alpha helix. So its backbone is like this, okay, like this, so in a zigzag manner, clear. And the arrangement of several segments side by side, all of which are in the beta conformation, are called beta sheet, clear. So all these structure that are basically arranged by side, note it down, that is arranged by side, and all of which are in beta conformation, we call it at beta sheet. A sheet like appearance is basically seen, so its name is basically formed a beta sheet, clear. And another important thing that I want to tell you, that hydrogen bond is formed. Just note it down, hydrogen bond is formed between the adjacent segment of the polypeptide chain within this sheet. Okay, so between the polypeptide chain, so if we look at this, this is one polypeptide chain and this is another polypeptide chain. So in between this polypeptide chain, hydrogen bond is formed. Okay, so between this polypeptide chain, hydrogen bond is formed in case of beta sheet, clear. So if we look at the difference between alpha helix and beta sheet, is that in case of alpha helix, hydrogen bond is formed, but in case of alpha helix, just note it down because this is important from examination point of view. In case of alpha helix, the hydrogen bond that is formed is basically intrachain hydrogen bond, intrachain hydrogen bond. Why it is intrachain? Because when I teach you alpha helix, I discuss about this, is that the hydrogen bond is basically formed in this same chain, that's why in case of alpha helix, the hydrogen bond that is formed is intrachain hydrogen bond. Okay, but in case of beta sheet, the hydrogen bond that is formed is interchain hydrogen bond. Okay, so why interchain hydrogen bond? Because in case of beta sheet, the hydrogen bond is formed between the two segments. For example, this is one segment and this is another segment. Okay, so here you can find out that hydrogen bond is formed between this one segment and another segment, between this. Okay, so in this way hydrogen bond is formed in case of beta sheet. So that's why, as the hydrogen bond is formed between two different segments in case of beta sheets, that's why it is interchain hydrogen bond. So this is the main difference in respect to hydrogen bond between the alpha helix and beta sheet, clear. Now next, see, so if we look at this strand, okay, that is beta strand. Okay, so beta strand is basically what? Monomer in nature, clear, monomer in nature. But when it basically form a sheet, then it becomes polymer, clear, then it becomes polymer. Okay, so these points are basically important. Okay, now another important thing that I want to discuss is that beta pleated sheet that is formed when two or more polypeptide chain segment line by line. Okay, so as I already discussed the difference between beta strand and beta sheet, so beta strand is monomer in nature, I already discussed. Okay, monomer in nature, while beta sheet is polymer in nature. Okay, but if I explain this in a manner, is that in case of beta sheet, or you can say beta sheet is formed when two or more when two or more polypeptide chains when two or more polypeptide chains when two or more polypeptide chains line side by side line side by side. Clear, students? So beta sheet is formed when two or more polypeptide chains line side by side, then only beta sheet is formed. That's why we call beta sheet as polymer in nature. Okay, but in case of beta strand, each individual segment of beta sheet each individual segment is called beta strand. That's why we call beta strand as monomer. So student, remember the difference between beta strand and beta sheet. Beta strand is the individual segment. That's why we call beta strand as monomer in nature. But when it forms beta sheet, there will be two or more polypeptide chain segments, which are basically lined side by sides. That's why we call beta sheet as polymer in nature. So this is a basic difference between beta strand and beta sheet. Now I hope that you get a clear idea about beta strand and beta sheet. Okay, note down all the things that I discuss here. Okay, next, see, as I already discussed that in case of alpha helix, it is basically coil in nature, but beta strand is what? Zigzag shape. And because of that zigzag shape, beta strand is quite extended. And if we plot it in Ramachandran plot, then we see that beta sheets are basically found towards the 180 degree due to its extended nature. Okay, I hope you note down all these things. Okay, now another important thing is that the distance between the adjacent amino acids. Okay, the distance between the adjacent amino acid along beta strand is 3.5 angstrom. Okay, so just note it down. Distance between the adjacent amino acid in beta strand is approx 3.5 angstrom. Clear? So this is a very important point, that is the distance between the adjacent amino acids along a beta strand is about 3.5 angstrom. Okay, while we see in case of alpha helix it is 1.5 angstrom. Clear? So this is another important point regarding the beta sheet, or you can say beta strand. Here we discuss about the individual segment. Okay, and I already discussed about the hydrogen bonds, that beta pleated sheets are stabilized by the interchain hydrogen bond. That is the main difference between the alpha helix and beta sheet in respect to hydrogen bonds. Clear? Now another important thing that I will going to discuss here is that beta strand, or we can say beta sheet, is basically two types. One is parallel beta sheet, one is anti-parallel beta sheet. Okay, so one is parallel beta sheet, one is anti-parallel beta sheet. Okay, so if both the chains start with same terminus, then it will be parallel beta sheet. Okay, so if we look at the beta sheet, parallel beta sheet, just draw with me. So this is a structure of parallel beta sheet. Suppose this is the amino terminal and this is also amino terminal. Okay, clear? So if both the chains start with the same terminus, that is ammonia, then it will be parallel beta sheet. Okay, so this is a diagram of parallel beta sheet. Clear? Again, clear? So this is a diagram of parallel beta sheet. Okay, and in case of parallel beta sheet, the hydrogen bonds is formed like this, okay, like this, okay, like this, okay. So this is the parallel beta sheet. So if both the chain start with the same terminus, then we call it parallel beta sheet, and if both the chain are start with different terminus, then we call it anti-parallel beta sheet. Now let's see the structure of anti-parallel beta sheet. Okay, so here the terminus is different. This is the amino terminus and this is the CO terminus. Okay, so in this case the structure looks like this. Okay, clear? So in this case the hydrogen bond is formed like this, this, this. Clear? So this is the anti-parallel beta sheet. As in this case, the chain start with different terminus. One is amino terminus and one is CO terminus. So this is anti-parallel beta sheet. So I write down here, this is parallel beta sheet. I just write out parallel beta sheet, and this is anti-parallel beta sheet. Clear? So this is a basic difference between the parallel beta sheet and anti-parallel beta sheet. Okay, so from this diagram you may conclude that which is more stable, parallel beta sheet is more stable or anti-parallel beta sheet is more stable. So the answer is that anti-parallel beta sheet is more stable. So why the anti-parallel beta sheet is more stable? Because if you look at the structure of anti-parallel beta sheet, if you look at the structure of anti-parallel beta sheet, that is this structure, so here you can find out that in case of anti-parallel beta sheet, the hydrogen bond that forms always in 180 degree angle, okay, you can find out that straight 180 degree angle. Okay, and we know that hydrogen bond, when we basically discuss about the non-covalent bonds in our first lecture of biochemistry, there I discuss is that hydrogen bond is more stable when it is basically 180 degree. Okay, so in case of anti-parallel beta sheet, the hydrogen bond that is formed between the two polypeptide segments is 180 degree, that is more stable. That's why anti-parallel beta sheet is more stable as compared to the parallel beta sheet, because in case of parallel beta sheet, here you can find out that like this, not 180 degree. Okay, so this is a basic reason that anti-parallel beta sheet is more stable as compared to parallel beta sheet due to the formation of hydrogen bond in 180 degree angle. Clear? Or you can also say that anti-parallel beta sheet are more stable than parallel beta sheet because fully collinear like this hydrogen bond is formed in case of anti-parallel beta sheet. Okay, so students, this is all about the beta sheet. Okay, so that's it for today's lecture. Okay, I hope that you like today's lecture. If you like today's lecture, then please hit the like button and please subscribe to our BioSmart Academy YouTube channel. In our next lecture, we will discuss the part 2 of today's lecture, that is about the beta turns. Okay, we will discuss briefly about the beta turns in our next lecture, as well as I will show you some good pictures of beta sheets. Okay, then you will get more clear idea about the beta sheets. Okay, so please share and subscribe to those students who require these lectures, because all these lectures are strictly made according to CSIR net syllabus and all these lectures are freely available. Okay, thank you very much.